Hello everybody, this is Dr. Kevin Connors, and today we're going to be discussing your gene profile. So these are people that have done this gene profile, and we're going to go over it together. You're seeing my computer screen here. So hopefully we're going to answer these questions when we go over this together. So I'm not going to go over every single gene in this 23andMe test, but we're going to go over some classifications of this. It's taking a long time to go over these profiles with people, so I thought, well, we need a little bit more education here. I know this is a learning curve, and there's a lot to learn, and trying to absorb it all in one sitting is difficult. That's why I thought putting together a video will be helpful for everybody. So let's go through this. What are we looking for in these genes? So this is everybody that's done the 23andMe profile and now have sent me the data. And once you send me the data, I put it into my little handy-dandy computer program, and we get a spit out that looks similar to this. Now I just took little chunkets of this, that you're going to get this sheet just like this. That's going to be about 11, 12 pages long. And we're going to look at each one of these individual genes, and I'm going to talk about these individual genes. Now this is actually somebody's profile that I took their name off of. So it's going to break it into different compartments. But before we get to that, I want to just reiterate some of the things that I've already talked about, just so you remember that when we look at your genes, there's some things that you need to keep in mind. Number one, just because I have a genetic defect doesn't mean I'm going to necessarily have a problem. Genetic defects can be present in everybody. Matter of fact, there isn't anybody that doesn't have genetic single nucleotide polymorphisms, and they can live to 120 years old and be just fine. Matter of fact, there's some SNP defects that can help prevent cancers, that can help prevent different things, that could be beneficial given different environmental circumstances. And there's some that can cause some issues. Now, all of them will cause more issues if there's environmental insults as well. Meaning that if I'm never exposed to Lyme or harmful chemicals or H. pylori or other bacteria or pathogens, etc., then my genes aren't going to really play a big part in my health. However, if I have gene defects, plus I'm exposed to Lyme infection, and I'm exposed to different pathogens, and I have heavy metal toxicity, and I was exposed to mercury in my fillings, then you have all these insults that lead to problems. So when we look at this genetic profile, it's important to understand that there are all sorts of environmental things that play into my health. Then we take a look at the environmental things plus the genetic issues, and it can tell a picture that can help us manage the patient. So specifically when we look at this gene profile, we're looking at, it says variants. So in this picture, in that little blue box, you can see my arrow here, we're looking at the HLA gene, and it says variants. In this scenario, we're looking at that it says 1 here, and none of them say 2 here, because this person only has a single allele defect in these genes, or it doesn't have anything in that box. So that would mean that this person inherited a normal, what's called a wild gene, from his mother and from his father, meaning he doesn't have any defects. In this first one where it says 1, that means this person inherited either a defective gene from his or her mother or his or her father. So this person has a single allele defect. So understand each one of these genes, this HLA gene for instance, can be a family of genes. In this case, there's a family of at least 2 genes that are measured here. The FUT2 gene here is a family of 3 genes that are measured here. Each one of the genes has 2 alleles, one from your father, one from your mother. If I have 0 defects, I can still have problems with the gene because of an environmental insult. If I have 1 defect, I could have more problems with the gene given environmental insults. If I have a double allele defect or two variants, I could have even more problems with the gene. Still doesn't mean it's not going to work completely, but it means that I could have more problems with it. All right, so let's jump into that so we have all those little caveats that you can remember. When we look at the HLA genes, those are the genes that are known to affect gluten, meaning that if I have one of those genes, like this person has a single allele defect on an HLA gene, that means they're going to be more prone to be susceptible to gluten intolerance. So if this was you, and I said you have a single allele defect on your HLA gene, it would be my suggestion that you go gluten-free if you're not already gluten-free. If you don't have any defects on the HLA genes, does it mean you don't have to go gluten-free? Again, the answer is no. It would still be beneficial to go gluten-free because gluten is inflammatory. So if you have genetic defects of the HLA gene, it's even more important to be gluten-free. Following me? So if a person was gluten-free already and they showed no defects on the HLA genes, the correct thing to do is to stay gluten-free. If the person is not gluten-free and they have some defects on the HLA gene, then it's even more important that they go gluten-free. Follow me? So the genes aren't the telltale for everything. The genes just give us an indication of what's going on. You'll understand it better as we continue. The second digestive factor gene is your FUT2 genes. Your FUT2 genes help make the slime layer, for a better sake of a better term, that coats your intestinal walls. So everybody knows about your flora of your intestines, and you need to take probiotics so that your intestines have a good, healthy probiotic layer, so you have that symbiotic environment of microbes that help you digest things and absorb things and kill off and fight off pathogens. So we take probiotics. That's helpful. Well, that's good, but where do the probiotics live? Well, they live in the gut. Well, where do they live in the gut? They live in the healthy slime layer that coats the intestinal walls. That healthy slime layer is made by the epithelial cells themselves through a process using this FUT2 gene. So, if a person has a defect, like this individual has a single allele defect in all three of those FUT2 genes, that's not so good. So they are going to have more difficulty making the apartment complex for the probiotics to live in. So this could be a person, and if this is you, you could be taking all the probiotics in the world and you can't figure out why you can't get your gut back in order. Somebody told me I had leaky gut. I've been taking probiotics for a long time and I still have gut problems. It just has never gotten back in order. I still have inflammation in my gut. My digestion's off. I just can't figure out why I can't get better. This is one of the reasons why we do these gene tests, because it gives us some answers to things. Well, then we look at your gene tests, and you got a single allele, or I've had people with a double allele defect in every one of the FUT2 genes. Well, oh my gosh, no wonder you got a bad gut. You got on top of that the antibiotics that you took, and eating genetically modified foods, and eating junk foods, and additives, colorings, flavorings, things like that, and you're toast. So what do you got to do? You have to heal the gut. So go back to my gut check video and understand that if you have any defects in your FUT2 genes, you need to be on prebiotics for sure. Most likely your whole life. Being on prebiotics, being on probiotics on a long-term basis. Now, take a step back. Let's say a person has no defects here. That's the person that they might have a messed up gut because they took antibiotics and because they had a poor diet, but it's easier for them to recover. So they get on prebiotics for a month or two, they start taking probiotics, their gut is normal, and they're feeling great. Pretty much guaranteed they don't have a whole lot of defects in the FUT2 genes. But a person that's, gosh, I've been working on my gut for the last year and I can't seem to get better, you probably got some defects. So you understanding how this all comes together a little bit? So it gives you some answers so you don't think you're crazy sometimes. So it can really help looking at your genes, and it helps you manage it, knowing that, wow, this person, have a single allele defect on all my FUT2 genes. I'm going to need to be on prebiotics and probiotics the rest of my life. I'm going to need to watch my diet more closely. I'm going to need to stay away from antibiotics. I'm going to need to make different health decisions that way. It helps you manage your life. Let's move on. Genetic variants that influence histamine levels. So there's 2 genes in your body that degrade histamine. Now understand, histamine is an immune chemokine made by your mast cells that is part of your immune system. And histamine is needed to help kill things and remove pathogens, but it's supposed to be secreted by mast cells, and it's supposed to be then degraded quite quickly, literally within a few minutes, by these specific genes. Your ABP1 gene family, and there's 1, 2, 3, 4, 5 that are measured here, in your ABP1 family, these genes degrade histamine in your gut. So, how would I get histamine in my gut? Well, histamine is an immune cell. The mast cell secretes histamine. Also, there's histamines in different foods. So foods create a histamine reaction. So let's go back to gluten a second. So if I'm going to eat a lot of gluten, when I have antibodies to gluten, I'm going to increase my histamine level in my gut, and I'm going to need plenty of healthy ABP1 genes to make an enzyme, the DAO enzyme, that degrades histamine in my gut and breaks it down. So I don't have this inflammatory process going on in my gut. Now, histamine is only one of the inflammatory chemokines. So don't get me wrong, just because like this person doesn't really have very many variants at all, doesn't have defects at all, I would say ignore your histamine issues to this individual. That doesn't mean that they're not making other inflammatory cytokines from a gluten sensitivity because they have gluten antibodies. So that's another piece of the puzzle. But in this case, this person has one single allele defect in one of their ABP1 genes. Not a problem. Guarantee you, this person does not have a histamine problem. The second gene, the HNMT gene, actually makes the enzyme in the body, in the extracellular spaces, so that it breaks down histamine in your body, because mast cells are releasing histamine in your body to kill pathogens all the time, and the HNMT gene is responsible to make the enzyme that degrades histamine in your body. So let's pretend this is you, and let's pretend in the ABP gene you have multiple defects there. So that would tell me that you do not make DAO enzyme that breaks down histamine in your gut very well at all. So then my recommendation would be you have to decrease the amount of histamines that you consume, and make sure that you're not consuming any foods that you have antibodies against. So a Cyrex panel at the lab would probably be in order to see if you have any antibodies to any different foods. Testing you with kinesiology, whatever we need to do to find out any sensitivities that you have to foods that you need to stay away from. And then looking at a list of foods that contain histamine. Eggs, fermented foods, nuts of all types, cheeses, things like that have high amounts of histamine in it, that is throwing histamine into your gut that you don't create the enzyme to get rid of. So you can't be eating foods that have histamine if you can't get rid of histamine in your gut. You will absorb it, you will cause this histamine reaction in your body, and you're going to have all sorts of problems. And that could be, so again, with a gene profile, you could go, wow, this is your reaction. This is why you're so hyper-inflammatory. This is why you have rashes. This is why you have heart palpitations. This is why you have gut problems. This is why you have migraines. Those are all symptoms of histamine. If, like in this case, this person has an HNMT gene, that is, his whole gene profile is pretty normal there. I mean, you could get much better than that. But let's say in your profile, you had multiple gene defects in the HNMT gene. You can't get rid of histamine in your body. So you better not be consuming histamine so that you're absorbing it and creating an increased histamine reaction. And you better find out that you're not and you have an autoimmune disorder, that you're going to be producing a whole lot of histamine as well. And you need to do some things, some nutritional approaches to degrade histamine extracellularly, meaning in the body, and get it out of your body. Does it all make sense now? Because too much histamine, everything in your life is a balance. Everything from a biochemical process in your body is a balance. It's all about this delicate balance. You need histamine. It's good for you. But you need to be able to get rid of it because it's bad for you if there's too much of it. So it's all about that balance. And if you have a genetic variant that doesn't allow you to keep that balance, then you need to help manipulate that balance so that you can bring health back. So it's, again, invaluable test. Moving on, genetic variants that may impact cell health and cell energy production. So this set of genes helps you get nutrients into the cell, break down fats and carbohydrates and proteins into end products to go through glycolysis and go through the Krebs cycle so that you can make energy. You could say that your one life's purpose, you have several life's purpose, right? Well, write this one down. One of your life's purpose is to make acetyl coenzyme A. So if next time you're in a class and you say, somebody asks you what your life purpose is, you say, well, it's to make acetyl coenzyme A. Later you're going to see it's to make SAM2. But acetyl coenzyme A is what comes out of glycolysis and feeds into the Krebs cycle and makes all sorts of ATP, if you remember from your biology class in high school, and that is essential for you to have energy. Now this person has multiple defects in that area. So in this gene that makes carnitine, ACT, and the SLC gene, and you can't see the ACS gene that I deleted, it couldn't fit on the slide down there. Boy, they got a mess of defects. So with this mess of defects they have, I can tell without even knowing this person's history, I can say this person's going to have chronic fatigue. They can't help but have chronic fatigue. So remember I started out this presentation that just because you have all these gene profile defects, if you have all these defects, it doesn't mean you're going to be a mess. However, if you got a lot of defects, you can kind of get a pretty good picture. So in this case, with all these defects, they're going to have a hard time making ATP. So granted, now if you add Lyme disease or you add cancer or you add some other dysfunction on top of that, it's going to accelerate this problem and just literally blow it up. And that's the case with this individual. They have a lot of symptoms because of that. So they don't make a lot of energy. So supporting this pathway with some specific nutrients is going to really help them. So then now we know. So now we know you don't make acetyl coenzyme A. We need to really help support that pathway, push that pathway, and really help that person with their symptoms. Does that make sense so far? All right, you still with me? I know this is a lot to go over, so you might have to listen to this a couple times. And we're going to go over yours specifically. So write down your questions. Next pathway, detoxification. Kind of important to detoxify. Remember, your level of toxicity is dependent upon your ability to detoxify. So if you are toxifying yourself at a rate greater than your rate of detoxification, you are going to be a sick person. So how do we know I can detoxify very well? Well, detoxification does have to do with your genetic picture. It has to do with other things too. Where's my stress level at? Etc. But we're looking at gene profiles here. So some of your detoxification pathways are in your SOD pathway. So the first gene that we want to look at, we're talking about detoxifying, is superoxide dismutase. I think I got another slide on this. Let's switch to this other slide on superoxide dismutase because this is kind of important, especially when you deal with Lyme patients. Superoxide dismutase is a gene aptly named because what it makes is an enzyme called superoxide dismutase. So the SOD gene makes SOD, and the whole purpose of SOD, superoxide dismutase, is to take superoxide and turn it into hydrogen peroxide, which then turns into water, and your body excretes it. What's superoxide? Well, superoxide is a byproduct of your body's metabolism. It is necessary. It's actually a free radical, but before you call it bad, remember, life is a balance, and you need free radicals in your body because free radicals can help kill pathogens. So this free radical, superoxide, kills pathogens, but you only want it in solution for a very short period of time, and we don't want that to build up. Well, it's not going to, because God made us with a gene called SOD that makes superoxide dismutase that actually will degrade any excess superoxide, turn it into hydrogen peroxide, which other enzymes turn it into water, and we pee it out of our body. Yay, we got rid of it. So it's this cycle, this balance of life that is so beautiful. The problem is what if I have a defect on my SOD gene? What if I have a double allele defect on all three of my SOD genes? Ooh, that kind of stinks. Why? Because I'm going to have a buildup of superoxide. That could be damaging to my cells, buildup of superoxide. But I'm sorry, it gets worse. If you have an excess of nitric oxide, specifically cytokine-inducible nitric oxide, it will bind to this excess superoxide and you get peroxynitrate. That's what's circled there. The ONOO negative is peroxynitrate. That is a very damaging free radical. It's very harmful to your cells, causes a lot of fatigue, causes brain damage, etc. This is the perfect picture of a Lyme patient. So this is what I see a lot of my Lyme patients have. Well, a lot of my Lyme patients are very ill Lyme patients. So they're very ill because they have lots of things going on. Well, we want to blame it all on the Lyme. Well, there's other things going on too. It's very common with a Lyme patient to see a defect in the SOD gene. So in the profile it looks like this. These are the SOD genes. This person has a single allele defect on all three of those genes, so they certainly have a decreased function of the SOD gene. So it's not making enough superoxide dismutase. It's keeping an excess amount of superoxide in the tissues, and then it's creating a lot of peroxynitrate. Why? Because see this NO, this is nitric oxide. With Lyme patients, Lyme is a chronic infection, so it's not just Lyme patients. With chronic H. pylori patients, with chronic subclinical viral patients, they're producing an excess amount of a specific type of nitric oxide, which there's three main types of nitric oxide. In a chronic infection, you'll produce an excess amount of what's called cytokine-inducible nitric oxide through cytokine-inducible nitric oxide synthase. This will be circulating through the blood. It will combine with the superoxide and make an excess amount of peroxynitrate. This is what can be very damaging, and this is what causes Lyme brain. This is what causes the achiness, the pain that moves around in your body that nobody can seem to understand. Most of these people, so you got two types of Lyme patients. I had chronic Lyme and I just took Kexclaw and I'm just fine. I don't know what's wrong with you. They probably don't have a lot of genetic defects that we're talking about here. Then you got the other type of person that's got a lot of genetic defects, especially in the SOD gene and some other that we're going to talk about later, and they're producing a ton of peroxynitrite causing symptoms. So we want to look at these things because you can support this pathway and you can really help this so they're not getting as much of this. There's products that can help scavenge peroxynitrite and grab onto it and get it out of the body. So knowing this can be invaluable to some of these chronically ill patients. It's just a great thing to know. So back here, looking at this gene profile, knowing you have these variants can be super, super helpful. Next one is glutathione production. See if it's in this picture. Yeah, kind of down in this corner. I kind of cut it off. But glutathione is recycled in your body. It's oxidized, reduced. You're recycling glutathione around this loop over and over again. And glutathione is your primary antioxidant in your body. So it antioxidizes a whole bunch of other chemicals and garbage that you got to get rid of. This person has 3 defects that are going to affect those. Now, they're single allele defects, and these are effector things, so not a horrible thing with glutathione for this person. I've seen much worse than that. Now, as we move down, the CYP genes are your cytochrome P450 pathways. Your cytochrome P450 pathways are essential. It's your primary detoxification pathway. This person has multiple defects in their cytochrome P450 pathway. They're going to be a slower detoxer overall. And then your PON pathway that's kind of cut off down at the bottom of this screen is at least right now the research is showing that's how we detoxify chemicals, multiple drugs, pesticides, herbicides, etc. And it's not uncommon that you see a bunch of defects in that PON pathway. People with defects in their PON pathway have to be very careful that they're making sure that they're buying the dirty dozen, foods that have the highest rate of or highest content of pesticides and chemicals and things that you should buy organic. You need to really be, really tight on making sure you're staying away from pesticides and herbicides and things like that if you have defects in your PON pathway. So again, you can see how knowledge can help make decisions in your life, and it can help keep you healthy. Doing pretty well so far? Not overwhelming you? It's not so bad, is it? So again, this is just a picture of some of those pathways. This is the part of the transsulfuration pathway right here. So this homocysteine is coming from the methionine pathway up here through CBS, is feeding down to glutathione, and what's out of this picture is the Krebs cycle and where you make your energy. Now let's move to the folate pathway. So if you're lucky enough to do this whole going over your test in my office, then I can kind of show you some charts and where these fit in, and maybe I'm the only one that finds this fascinating, but oh well. So your folate pathway, the purpose of your folate pathway really is to make methyl tetrahydrofolate. And maybe one of the most researched of these genes is the methyl tetrahydrofolate reductase gene. That's a family of several genes. The most important is the one right down here, the MTHFR C677T, and that's probably one of the more researched genes. And this poor soul has a double allele defect of that gene. Now, the research on this gene, the MTHFR C677T, if you have a double allele defect of this gene, it's going to slow your detoxification processes about 60 to 70%. Now that's a very important thing to know. This is where we get into some philosophical questions like, should I vaccinate my child? Well, if I knew my child has a 677 double allele defect, there's a way you should vaccinate your child. So I don't care if you believe that vaccines cause autism or not, but using medication as the mother, if you knew that you had an MTHFR defect and know that since you have a double allele defect, your child has at least a single allele defect. If you knew what your gene profile was of the father was, then you'd have a real good idea what the child's is. At least you could do a little graph and figure it out. You better this really should help you make some better decisions. Because, okay, so here's a mom. Let's use this scenario here. A mother does her gene profile. She knows she has multiple cytochrome P450 pathway defects in some of these other pictures that we saw, some PON defects. She has an SOD defect. She has a double allele defect of her MTHFR. There's no way I'm taking medications while I'm pregnant. You got to figure out a natural approach to do this. And there's no way you're going to take medications delivering this baby. Because you are setting this little 6-pound baby up for all sorts of disasters, because they're not going to be able to get rid of things out of their body, meaning drugs, chemicals, vaccines, etc., very well at all. Period. And these are the kids that end up having brain issues because of choices, that medication, vaccinations, etc., versus a child who has got zero defects of this. They could get vaccinated and it goes right through them and they, pee it out in their diaper. There's no issues whatsoever. So, do vaccines cause autism? No. But do vaccines and chemicals and additives, colorings, flavorings, everything, can they cause autism? Well, if you are that slow of a detoxer, it's going to be circulating through your body. It's going to cross the blood-brain barrier, especially in a child where their blood-brain barrier isn't even fully formed for several years. And you think that's not going to cause problems in the brain? You are dreaming. So knowing these genetic profiles can really shape the decisions you make. This is science. This isn't some philosophy of a natural doctor or something like that. Anybody that denies this is denying a lot of research. So you have to be kind of have your head in the sand to not deal with these issues. All right? So this is your folate pathway. You eat good cooked leafy greens. It goes through these genes all the way down here. The MTHFR gene is the one we were just talking about. Its main purpose is to make 5-methyl tetrahydrofolate. Feeds into the MTR gene, then feeds this way. That's not on this picture. That's the methionine cycle. Goes through the cycle of MTR, MTRR, which recycles B12. Feeds into the methionine cycle that makes SAMe. This methyl tetrahydrofolate then feeds down this way to the biopterin cycle, and this is constantly going on in your cells. This is feeding your cells. Little side note, so I don't have to repeat it when I'm talking to you in person, make sure absolutely you never, ever, ever take folic acid. Now, you can never, ever get away from folic acid because they put it in so many foods. But certainly don't buy vitamins that have folic acid in them. Folic acid is synthetic folate. Folate is what is found in green leafy vegetables. Folic acid is what's created in a laboratory. Folic acid is hypothesized to be actually one of the causative factors of a methyl tetrahydrofolate reductase defect. So the defects that we were talking about here, like this person who has a double allele defect in the MTHFR gene, can be caused from consuming folic acid. Now remember, go back to some of these other genetic presentations that I've done, that these are epigenetic defects, that some of these defects are believed to have been in the gene pool for thousands of years, and some are protective. Some have to do with environmental issues that our ancestors were exposed to. These are epigenetic defects. Some, like this MTHFR defect, is believed to be caused because of synthetic folic acid being introduced in the 20s and 30s, and it's still in vogue today, even though there's literally hundreds, maybe thousands of research articles sharing the danger of using folic acid. Still, it's in multiple supplements. So it's just, You stay away from it like the plague, all right? Next, all right, so now we're looking at the MTR, MTRR pathway, the way you recycle B12. So this MTR right there came from right here. Folate in your food, you reduce it to methyl tetrahydrofolate. It goes back to feed this pathway. It goes on to the biopterin pathway. This is what we're looking at now. Feeding in right there to the methionine pathway, another life purpose you have that you didn't know about, to make SAM. SAM is your main methyltransferase for your entire body. So that is what helps you make new cells, create DNA, and methylate. When your methylation process within the cell helps you turn on and off genes that you don't want to be on, or you need to turn on, you would remove the methyl group. So very, very, very important. So in this case scenario, this MTR gene, this person has a single allele defect. The MTRR gene, this person has a single allele defect. These are all the genes that feed into factors of a person needing help with vitamin B12. Why did we add the FUT2 genes here? Because that person had a single allele defect in all those genes. Why? Because where do you get vitamin B12? Well, you're supposed to absorb it through your gut, right? Well, where do you get it? Where you get it is from red meat. So you eat red meat that has good vitamin B12 in there. It binds to intrinsic factor in your stomach, and in your gut you absorb it. Well, if I have defects in my gut's ability to create a good mucosal layer, then I'm not going to be a good absorber of B12. Plus I have defects in my MTR and MTRR gene, so I don't recycle B12 very well. That kind of tells us that this person is going to need to be on a B12, most likely to help them, not only help them absorb it better, but help them recycle it. So they're on a cobalamin, methylcobalamin, adenosylcobalamin, hydroxycobalamin, depending upon their symptom picture. That's what's necessary to feed in to methionine. Let's move on. Then there's the PEMT and the BHMT gene. Go back to this picture right here. This is the BHMT gene right here. So as we move through the cycle and you make methionine, again, that has to go through the MAT gene, which we're going to talk about in just a second, in order to convert to SAM. So SAM then is your main methyltransferase for your entire body. Then it converts to SAW because it's all a balance. Oh, we got too much SAM, we're going to make SAW. That goes through the AHCY gene to homocysteine, and then homocysteine is shunted up. It's recycled again through the BHMT gene to methionine to make more SAM. And it's going in this pretty little circle. We're making SAM as we need to make SAM. Oh no, we have a defect. Well, we don't recycle homocysteine very well. Because why? Because we have a BHMT defect. Matter of fact, we got a double allele defect in 2 of the 4 genes. Oh no, that's not good. We're not making SAM very well. What's going to be the symptom picture of this person? Well, it could be more fatigue. This could be more even like brain fog fatigue too. There's a whole bunch of symptoms that go along with SAM, so I don't want to simplify that. But we have to talk about that with this person and how can we support the BHMT pathway. Again, we can't change the genetic picture, but we can help support this pathway with nutrients that help push it. Does that make sense? And that's not the only picture. We got to look at other things that can block it. Does this person have a heavy metal toxicity? Do they have a cortisol issue? Do they have a lipopolysaccharide? Well, what do they do? They have a lipopolysaccharide issue because they have chronic Lyme. So we got to kill the chronic Lyme too. Isn't this fun? Well, not if it's me. That's what you're probably saying. The next, the GAMT, AHCM, and the MAT gene. We started talking about those. This may be the most important, this MAT gene. They certainly is for this individual because they have a double allele defect in 1, 2, 3, 4, 5 of the 8 genes. Oh, that stinks. Yes, it does, because they don't convert methionine to SAM. And we already saw that they had a defect down here at the BHMT. So even if they are converting some homocysteine to methionine, it's not really getting to SAM very well. Ugh! Ah, definitely need some magnesium. Definitely need some ATP. Of course, they don't make ATP very good. No wonder this person isn't in my office, and no wonder they feel so cruddy. Gets kind of frustrating, doesn't it? Well, this is what makes my job so difficult, because there's so many different things to fix on this person. And if you ignore them to your own peril. So you have to look under every stone to figure this out. So that's what's so important when you deal with these patients. You got to look at the big picture, as big a picture as you possibly can. All right, so that's that gene profile. Now we move into the neurotransmitters, and this is where it gets really fun. Are you having fun yet, folks? Isn't this exciting? Boy, this has gone a long time, and I'm trying to talk fast. Oh, all right. So let's wind it down here. Let's talk about your neurotransmitters. So everybody thinks about the brain with neurotransmitters, but you'd have to have neurotransmitters everywhere, right? Because everywhere your nerves come in contact with another nerve, that's how they communicate, through these little chemicals we call neurotransmitters. So let's talk about these. First one, serotonin. So serotonin is what? Your happy neurotransmitter? Well, it's much more than that. But serotonin is degraded through MAO. Let's look at it on this picture here. This is your biopterin pathway. Isn't it pretty? So I have to shout out to Dr. Ben Lynch, who made these pathway planners. Great guy. Thank him for these beautiful pictures. I like pretty pictures, like anybody else. So this is biopterin recycling. Hopefully you can see my cursor here. Biopterin goes down through phenylalanine, tyrosine, tryptophan, and it feeds down to serotonin down here at the bottom, and you can see the MAO down here. MAO, M-A-O, is how you degrade serotonin. It is a Y chromosome defect, so men can only have a single allele defect. Women can have a double allele defect. And typically, we think of that defect, we think a lot of these defects as blocking that pathway. So, and that is the right way to think most of the time. So, but in lots of cases, it can either block or speed. In this case, it can do that. So serotonin is degraded through MAO. That's how you get rid of serotonin. You need to make serotonin. We need serotonin, but we have to degrade it. We can't have too much serotonin. So we have this enzyme called the MAO enzyme, which then degrades serotonin. However, if I have a MAO defect, I can either degrade it too quickly, so that I have too little serotonin, or I could degrade it too slowly, and then I have too much serotonin. So what happens in most these people with these defects, they have times that they have too little, and they have times they have too much. And here would be my symptom picture if this was me. If I have too little serotonin, I'm sad, and I'm maybe even a little depressed. And I just have some melancholy, and I have some sadness. If I have too little, I'm a little irritable, and I'm a little quick-tempered, and I'm a little short with people. So I could tell a lot about you when I look at your gene profile. Because the next gene we talk about is dopamine up here, and the COMT gene, the COMT gene. So dopamine, hopefully you can see my cursor, it's in the upper right-hand side, you see dopamine, and that's degraded through COMT. Also norepinephrine is, also through MAO, norepinephrine, and epinephrine is degraded through COMT. The COMT gene can do just the same. It can speed or it can slow, but it usually slows the degradation of that neurotransmitter. The COMT defect is much researched with ADD, ADHD kids that are seen to have in common to have a COMT defect. So they have an excess amount of dopamine. They tend to be a little bit more aggressive. They tend to be very stuck on things. It's hard for them to focus. It's hard for them to stay focused. They get distracted easy. They're impulsive, and they can be OCD. So that is the picture of a comp defect patient. So you add a comp defect with a MAO defect and a GAD defect. GAD is how you get rid of or how you create GABA. Now you add a GAD defect, so GABA is actually a calming neurotransmitter. It brings calm. You need a lot of GABA to go to sleep at night and rest. A person that has a healthy GABA production is calm and understanding and can talk through things. When I have GAD defects, I'm quick-tempered, I'm angry, and it's really hard for my brain to chill out and calm down, and I tend to be prone to anxiety. So here's a person, this individual, doesn't have the MAO defect, but I've had patients with a double allele MAO defect, 3 comp defects, and multiple GAD defects. I can pretty much tell you their temperament. So this gives you some answers. Then you look at the pathway and you go, well, how do I fix this? Well, how you fix this a lot is by tempering the diet. Now I have a whole paper I wrote on how to balance the comp and MAO defects, and I'm constantly adding to it. So it's just a way to look at, you look at what feeds it, phenylalanine, tyrosine, tryptophan. These are amino acids. Maybe it's not really good if I have a comp and MAO defect to be eating a ton of chicken and turkey and fish and meat and such that are going to add these amino acids. At least don't eat them without eating a lot of fibrous vegetables. Maybe I need to cut down on the meats a little bit and eat more vegetables. I might do much better. Still with me? Because it could get more complicated. You look at this whole pathway, this IDO pathway over here. This is what I call the tryptophan steal, especially in the presence of lipopolysaccharides. What's that? That's a byproduct of the breakdown of gram-negative bacteria like Lyme disease, the Burgdorferi bacteria, the H. pylori, INF gamma, TNF alpha. Those are inflammatory cytokines. So in the presence of chronic inflammation, stress, emotional stress, tryptophan gets stolen down the IDO pathway. This is the kynurenine pathway. Kynurenine forms eventually quinolinic acid, which can be an excitotoxin. And this can cause depression, inflammation in the brain, all sorts of problems. So this is where you get an autoimmune reaction, this ramped up inflammation in the brain and damage to neurons and glial cells and brain fog. So it could kind of be a mess, right? So in cases like this, we really have to watch the proteins that we're consuming and the amino acids that we're consuming, that we're not feeding this whole pathway. That if this person has a MAO defect and they're not degrading serotonin and they have a lot of aggression, we don't certainly want to be producing more serotonin. Look at the bottom of this, the bottom right-hand side, melatonin. Ooh, that's a byproduct of serotonin, but it's not degrading very well at all. We got excess serotonin. It's feeding back up here. We got the ADO steal, the tryptophan steal going on, making quinolinic acid. I'm not making melatonin. That's why I can't fall asleep. You see how this answers a lot of questions? And you thought you were just crazy? Well, you might still be crazy, but at least you know why you're crazy now. So this genetic pathway, properly understood, and understand I know nothing compared to what I hope to know in a year, is it really can answer some questions on why I have symptoms and what's going on. Is this helping you? Then you have the NOS gene that degrades and creates nitric oxide. Remember I said there's 3 forms of nitric oxide? There's NOS1, or what's called cytokine-inducible nitric oxide. That's what's ramped up in a chronic infection. What's ramped up in an acute infection too, but then it's supposed to degrade. But when I have a chronic infection, then I have this constantly ramped up nitric oxide, cytokine-inducible nitric oxide, or NOS1, which can bind to superoxide if I have a SOD variant and cause peroxynitrate problems, right? That's not good. And then there's NOS2, which is essential and important. Now understand, NOS1 is important and essential, but we don't want to have excess amounts of it in a ramped up chronic infection state. So again, everything is balanced. NOS2 is found in the brain, and that's protective and neuroprotective to the brain. NOS3 is also called endothelial nitric oxide. The endothelial cell is the cell that lines your blood vessels. That is absolutely essential to your blood vessels and the health of your blood vessels. And a deficiency in NOS3 or endothelial nitric oxide is your cause of high blood pressure. It leads to cardiovascular disease. You add that to these additional cardiovascular support genes, the ACE gene. This person has a defect in the ACE gene. This person is really going to be prone here to high blood pressure and cardiovascular disease and needs support in that area. So this very person, I can't remember, but they could say, well, I don't have high blood pressure. Well, you don't have high blood pressure, you don't know you have cardiovascular disease, but you definitely don't make as much endothelial nitric oxide, and you have issues with your ACE genes that make the receptor that will decrease your blood pressure. We certainly want to support that arena now before you end up having a problem. Does that make sense? So knowing these things prior to having a disease can be really helpful and help you make decisions to help monitor your life. This next gene is also really, all these genes are important, but this one is really important, especially with my cancer patients. This vitamin D receptor gene, this VDR gene, there's 3 genes that we measure in this gene family. And this person is pretty shot here. They got a single allele defect in 2 of them and a double allele defect in 1 of them. So this person does not make that, they have a difficult time getting vitamin D where it's needed, into the cell, into the bone. They don't have as many receptors. Remember, the vitamin D receptors also function as an apoptosis receptor to help prevent cell not going through apoptosis, which leads to cancer. So I want my patients that have this gene defect to have blood vitamin D levels, if they measure their vitamin D levels, certainly over 80. Matter of fact, if you had it over 100, I'd be much more happier, because they need to have much higher circulating vitamin D levels, blood-tested vitamin D levels, because they have a receptor defect. So with this gene defect, you want to really up your vitamin D levels. If you haven't gotten it tested, you can get your vitamin D blood tested. It's not a very expensive test. But this person certainly needs to be on 5 to 10 to 15,000 IUs of vitamin D a day. Is this helpful so far? All sorts of things to learn about your gene defect. And then this, my computer program puts this, this BH4 is biopterin. Biopterin has to do with your mood, has to do with your nitric oxide too. So let's go back to this pathway here. Biopterin recycling. So biopterin is recycled here, and it moves around this cycle like this, feeding, creating neurotransmitters, creating nitric oxide. See, this is ENOS, that's NOS3, same thing, endothelial nitric oxide here. And it's so important for your heart, it's so important for your neurotransmitters, it's important for your brain. It's just really important, isn't it? So defects in that affect your mood. It also affects your heart. So important cardiovascular features. Still with me? Isn't this fun? Well, we're at the end. So at the end, then after I go through all of this with you, I figure out, based upon your ratings on all those genes, a protocol for you. So your protocol is based upon those genes. I decide what we need to do to support those pathways. I can't change the genes, but we can certainly support the pathways. It has to do with your personal symptoms as well as what's going on with your gene profile. So in this case, we're going to use a product called B-specific, and a B-specific, that specific vitamin has your B support without the methylation support, because we can't support methylation first with this person because they have so many defects in the biopterin pathway that if we push methylation too hard, this is what we see a lot with patients. They come in and they're taking something that, because they ran one gene test and they have a MTHFR gene defect, so the doctor put them on high doses of methyl tetrahydrofolate. And that's not a good way to do it, because if you have other gene defects, now because they had all that circulating 5-methyl tetrahydrofolate, it pushes everything else. And this person had a ton of defects in the biopterin pathway. You can't add methylation support right away. So we're supporting their cell health because of the issues with fatigue and the issues with their ability to make energy. That's why we're giving mitochondrial support. You need minerals for all those pathways, and we're supporting SOD because one of the biggest problems this person had was brain fog and brain fatigue from excess peroxynitrate. So remember, that's from an SOD defect, so we have to support the SOD. And I could have used a peroxynitrate scavenger with this person too, but I don't like to use too many supplements at one time. And we just want to make sure things are going, and we have to tweak this and play with it. It's not something that they're going to be on the rest of their life. It might be they're only going to be on for a month or two, and we need to change things. So what's hard when you have genetic defects, it's affecting so many pathways, so you have to tweak the program. It's not like here's this cookie cutter thing. It just doesn't work that way. So you have to be patient with yourself, and that's how we'll all be happy, because you're a person and your body's changing all the time. So if you have questions, and if you're watching this video and you don't even know who I am, my name is Dr. Kevin Connors, and that's our website, Connors Clinic, up there. So if you somehow stumbled across it and stomached your way through it, I congratulate you. If you want more information about our clinic, you can contact us. If you're watching this video, most likely I made you watch this video, and if you're still here, I will give you a cookie because you watched the whole thing. So, good job. If you need to watch it a couple times, do so. And I look forward to talking to you about your test. All right, thanks. Bye-bye.